Alrighty, it's been a while since we've had a video lesson, so let's go ahead and talk about signal transduction. So, signal transduction is a process by which messages received from outside the cell, usually through a signaling molecule, which is called a ligand, sometimes pronounced ligand, and by which that signal received from outside of the cell is going to be transferred to the inside of the cell and then set into motion some sort of cellular response. And so this is a process that is very similar across very many branches of life, everything from bacteria all the way to animals and mammals. So that suggests a very early evolutionary origin for this type of pathway. There are three major steps: reception, transduction, and response. And we're going to talk about each of those in more detail. But first, let's talk about a few of the commonalities between some of these um, steps in the process. So there's a lot of steps that may involve protein modification, and these modifications are a lot of times going to be adding a phosphate group from ATP. This is called phosphorylation. A lot of times that serves to change the activity of a protein, whether to turn it on or to turn it off. And this is performed by protein kinases. Kinases add a phosphate. The opposite reaction of removing that phosphate is performed by phosphatases. And then you can have something called a phosphorylation cascade, where you have one enzyme phosphorylate another kinase, which phosphorylates another kinase, which phosphorylates another. And this goes on and on until whatever cellular response is triggered. This is a common site where cells amplify this signal, so that way you could have one ligand coming in activating one receptor and still generate a much larger response in the cell. So here we're going to look at a phosphorylation cascade. You have a bunch of inactive molecules, you have some ATPs, and you have a receptor sitting in the cell membrane. The ligand comes and binds, and that activates a second messenger. That second messenger can then in turn activate the kinase that's there, that first inactive kinase, which can then take ATP and make ADP and use one of those phosphates to activate another kinase. Now, this step can happen over and over. You can have one kinase activate another, but eventually you're going to trigger some sort of response by activating maybe another relay molecule, a transcription factor. We'll talk about those later, and that's going to trigger a cellular response. So this is what a phosphorylation cascade looks like. It's just a bunch of things phosphorylating each other, one after another. So let's talk about each of these steps in detail. Step one is reception, where a ligand molecule is going to bind to a receptor in the target cell. A ligand is a chemical messenger. It's usually either going to be water soluble. Some of them are not. They're hydrophobic. We'll talk about those in a little bit. And this is usually a very small molecule. It could be a peptide, something a couple of amino acids long. It binds to a specific site on the receptor, which then initiates a change in conformation, which changes the intracellular or inner surface of the receptor, which can then trigger all of these activities inside of the cell. Receptors are of two major types. You can have one that's inside of the cell, that's an intracellular receptor. You can also have a transmembrane receptor, which is in the cell membrane. So this right here represents the cell membrane. So you can see that it's stuck through the cell membrane. And a receptor is going to fit with its ligand, kind of like an enzyme fits with the substrate. Very much lock and key model with matching 3D structures. To talk about these, we're not going to focus on the intracellular receptors very much. I just wanted to mention that if you have a hydrophobic ligand that is able to cross the cell membrane. So the plasma membrane is permeable to these molecules. Very common, these are steroid hormones. They can pass through the plasma membrane, and so their receptors can be inside of the cell. Some of them can even be inside the nucleus. Let's talk about types of receptors. We're going to talk about two major types. The first one are called G protein coupled receptors, or GPCRs. This is a large family of proteins. You have a lot of GPCR types inside of your or on the surface of your cells. They work with the help of something called a G protein, which is called that because it's bound to either GDP or GTP. GDP is guanosine triphosphate, and GDP is guanosine diphosphate. It works very similar to ATP in that you can hydrolyze GTP to make GDP, and an inorganic phosphate. You can also perform the opposite reaction to add a phosphate to GDP to form GTP. While this is sometimes used as an energy storing molecule like ATP, it's very rare in the cell. A lot of the places where we'll see GTP this year is basically just in these cell signaling pathways. Okay, so what ends up happening is this G protein is an on-off switch. If it has a GDP bound to it, it is inactive. You can think of it almost like the GDP is lower energy, so it's going to represent something turned off. If it has a GTP bound to it, the G protein is active. So you can think of it as higher energy, means the G protein is turned on. What's going to happen is the receptor is going to activate. It's going to cause a G protein to trade out a GDP for a GTP. It's going to turn on. And then that G protein can go on to activate some other molecule, a lot of times enzymes or kinases. And then once it's done with its task, it's going to hydrolyze that GTP to GDP, and then it turns itself off. So this is kind of my animation. There are a few inaccuracies. So for example, it's just a little hard to change at this point. This is a G protein right here, this green molecule. It should be bound to that GPCR, but let's go ahead and have the ligand bind. That's going to cause the G protein to switch out its GDP for a GTP, and then it is going to become activated. Once that happens, that G protein can then move on to activate a molecule, a lot of times an enzyme, and that's going to trigger a cellular response. After that, once it's done its task, the G protein will then hydrolyze its GTP to GDP, at which point it turns off and it can go back and bind to that G protein. Also, once the active G protein has been dissociated from the enzyme, it becomes inactive. The second type of receptor we're going to talk about is a ligand-gated ion channel. What this is the ligand acts like almost like a lock to a door, and it can open and close that door. That allows this ion channel to change shape, which allows ions to pass through. A lot of times this is going to be ions, positive ions like sodium or calcium. Sometimes it'll be something like potassium or chlorine. This is very, very important in the nervous system and the muscular system as well. So if we look at this, a ligand binds. That pretty much opens a gate which allows ions to pass through. They tend to diffuse down their concentration gradient, so this is going to be passive transport, not active. And once the ligand dissociates, it's going to allow the receptor to close, the ion channel closes, and ions can no longer pass through. So it's a gate that you can then open and close. Now, let's talk about a specific category of ligand. These are hormones. Hormones are molecules, signaling molecules released by glands in the endocrine system. They are usually going to be carried in the bloodstream to their target cells, and they are capable of lasting for and traveling long distances in the body. So you can have a gland, for example, the adrenal gland, which is located on top of the kidneys in your lower back, can release adrenaline, and that travels through the blood and can reach pretty much every cell in the body and have an impact. Let's talk now about what happens inside the cell. So signal transduction is passing the message on inside the cell, and this is done by second messengers. Second messengers are small water-soluble molecules. They have to be water-soluble so they can float around in the cell. They diffuse, so they're not actively carried anywhere. And a lot of times they're activated by those GPCRs we talked about. Some examples are cyclic AMP and calcium ions. Those are the really big examples. I'm going to put inositol triphosphate or IP3 up here, even though it doesn't tend to show up very much on the test, just so if you see it, you know, that's a second messenger. So calcium ions can act as a second messenger, or they can be released due to the activity of one of these cell pathways. Notice that you can have pumps that are actively using ATP to pump these calcium ions to the correct concentration gradients. That way, when an ion channel opens, calcium can flow either into the cell or into some compartment in the cell. Calcium, when it gets into a cell, is able to trigger a bunch of different pathways inside of the cell to do a lot of different functions. So some cells find this calcium so important that they have an internal storage, usually a modified bit of endoplasmic reticulum, that stores extra calcium that can then be used later to release calcium into the cell. Cyclic AMP is a really big second messenger. It is made, as is shown down here, you take ATP, you change it to basically you change it to AMP, which means you pop 2 phosphates off of it. So adenosine monophosphate is then turned into it's called cyclic because it forms a circle. It's hard to see in the diagram. This is basically the circle right here that you're forming. And cyclic AMP is produced by an enzyme called adenylyl cyclase, which is up here. Adenylyl cyclase is often activated by a G protein and then will form cyclic AMP, which usually goes on to activate protein kinases, which phosphorylate other proteins. So when we look at what happens inside of a cell, we can see something called amplification happen. This happens during signaling cascades. So when a message is relayed from the receptor all the way to whatever's going to affect the cellular response, the response can be amplified at each step. So here in this picture, we see a receptor activating some molecule, could be a second messenger, could be an enzyme, which can then go on to activate multiple molecules, in this case enzymes, each of which can go on to activate multiple second messengers, and so on and so on. So we see at each step the reaction gets amplified or gets bigger. Let's talk now about how cells respond. So when cells respond, there are a number of different possible output responses. They could modify proteins either by methylating them, adding a methyl group, or by triggering a phosphorylation cascade or phosphorylating certain proteins. They can induce apoptosis, which we'll talk about in more detail in our next lesson. They can cause the cell to grow or to move in a certain way, cause the cell to release chemicals, or what's most common is to activate a transcription factor and modify gene expression. So turn on the expression of a gene or turn off. So gene regulation is a very common result. What happens is these signaling molecules will then activate a transcription factor, which we'll talk about more in the next unit. A transcription factor is going to usually encourage the expression of certain genes. So once you activate a transcription factor, it's like turning on a gene so that a specific protein will be made. Now, all of this has to be reversible because a signal is only useful when you can turn it on and off. So at every step of the way, there's a way to undo whatever happened during this signal transduction. So you can very quickly terminate the response by the ligand unbinding or dissociating from the receptor. A lot of these molecules in the signaling pathway will reset themselves. So G proteins, once they hydrolyze their GTP, they are now bound to GDP, and they are capable of being activated again. Ion channels, even if a ligand is bound, a lot of times they will close after a certain period of time. And proteins in the pathway that were phosphorylated can then be dephosphorylated by phosphatases. All of this resets the pathway so the cell can get a new signal anytime it's needed. Another interesting thing is that the same ligand can have a different impact in different cells. So here we see a ligand binding to a receptor, activating some second messengers, and initiating a certain response. But if we have inside the cell different second messengers, even if the receptor on the surface is the same, this can trigger multiple responses. In fact, you could have a single ligand trigger multiple responses within a cell because it activates multiple pathways. Or you could have a receptor that is completely different from the receptors in the other cells, and that then triggers a separate response. So you could have the same ligand trigger different responses in different cells, which is important when you have something like a hormone. You don't want every cell to do the exact same thing. You want each cell to do its targeted response. All right, we'll talk about this more later, and hopefully more lessons and videos will come soon.